This is the second part to the replication tutorial. Now this is the slide we stopped on last time, so we're continuing from that point. We were looking at the replication bubble and the direction of replication. Now remember that anytime you're starting replication, you are starting from the middle point. Now if we move to the bottom figure, you can see the most recently synthesized DNA is always going to be at the edges near the replication fork at the ends. This is because you always start the replication in the middle. So remember, replication would be starting in the middle point here. Now, we kind of discussed that because replication happens in the 5' to 3' direction, that you always have to worry about the newly synthesized strand becoming in the 5' to 3' direction. Now last time we focused on the right-hand fork, which just basically means it's to the right-hand side of that center line. We didn't really discuss the left-hand fork, which is to the left of that center line here and here. But now if you notice, this is going to generate a situation where you can have different events happening on the right-hand side and on the left-hand side. So let's take a look at the top strand. So that would be this strand here. That is our template with our 5' end over here and the 3' end again over here. So if you start the very first replication would go to about right there. Okay. Now, we talked on the previous tutorial that is why the first arrow on the right-hand fork would go from 5' to 3' in that direction. Now, it would also occur in the same direction. For example, on the left-hand fork, it would still start here, where I'm drawing the circle, and go in that direction. But now something would happen. You would now have the second part of the strand opening up, which would go to about right there, where the red lines are, dotted lines. Now, a slightly different thing would happen. If you look at the left-hand fork, you could just continue replication. It's already heading in that direction, and you can just continue on until you get to the red line. The problem is with the right-hand fork in this particular case, you are also heading in that same direction. But now you need to replicate the area outlined by that little arrow. Now there's a couple choices. In theory, what you might think is it kind of just flips around and replicates in that direction. But obviously that would be violating the 5' to 3' rule. So it doesn't just kind of turn around and replicate in the other direction. What you have to do is you have to start all the way over. You have to start back here at the 5' end of that and go in the proper direction. That's why the arrows are shown in smaller little segments. Okay, because the replication occurs in those small little segments. As you keep going, the same thing will happen. So the entire right-hand fork of the upper strand is going to occur in those little segments. However, if you notice the left-hand fork of the upper strand here, the top template strand, it can keep continuing because it's always going in the correct direction. So here it looks like a continuous strand. This leads to specific terms. which you may remember from before, what is known as a leading strand, and it's written there as well, and the lagging strand. The lagging strand has to synthesize the new strand in fragments, like you see here, all of these little tiny fragments. The leading strand does not need to create all those small little fragments. It can do it continuously. Because of that, the leading strand is called the leading strand because it can occur faster. It can just keep going. The lagging strand would lag behind in synthesizing because it has to kind of start over again every time synthesizing each of those little fragments. Now the other thing you should notice, and we can use the bottom strand that's not been kind of manipulated at this point, to see something else. The bottom strand here, the bottom template, if you notice, the right hand fork here is continuous, and the left hand fork has those little fragments, so is the lagging strand. On the same template strand, meaning this strand here, on that same template strand, you can have both a leading and a lagging strand. Here, the leading strand would be this, the lagging strand would be here. So on the same template strand, you can have a leading and a lagging strand. The same is true, obviously, above. Therefore, you can't have for a certain template say that it's always going to be a leading strand or an always lagging strand. It doesn't really work that way. Now for the DNA, when we're synthesizing it, we have kind of gone over roughly how it occurs, creating leading and lagging strands. But there's other components that we need to add, little details, to make sure that everything is explained properly. First of all, how does the replication bubble open up? How do you get from a double helix, which is connected by hydrogen bonds together, to separate so that you have the ability to carry out replication? 2 enzymes are needed for that. I shorten mine a little. The first enzyme is called helicase. Helicase separates the 2 strands of the double helix. So here it's called opening up the double helix. That's its job, is to make sure the hydrogen bond between the 2 double strands is separated. And now you can get all the enzymes in there to carry out replication. However, when helicase does its job, right over here, where they're kind of showing this circle arrow here, right where that occurs, that develops tension. Imagine if you had cords kind of wrapped up together and you were trying to pull them apart, and they by taking the middle and kind of just pulling it apart in the middle. What tends to happen is that the 2 ends around that opening where you've created, there's a lot of tension there. That can end up damaging the DNA, and you don't want that to occur. So there is another enzyme called topoisomerase that reduces that stress that builds up when helicase does its job. Now, how does topoisomerase work? Well, imagine if you had 2 strands of DNA and they are connected by hydrogen bonds, which are the dotted lines there. And let's just say there was a lot of tension there. Even though I'm not a good artist, so I can't represent tension very well. But imagine there was a lot of tension in this area of the DNA strand. How might you relieve that tension? The problem is because it's a double helix and you have these hydrogen bonds here, that actually inhibits the ability to release the tension. But instead of breaking the hydrogen bonds, there is another option that you can do. What you can do is break one of the backbones on one of the DNA strands, just one. Okay. And what that will create is, after you've cut that, you'll have this. Now the hydrogen bonds are still there. But notice what you have is you have this cut or a separation. That allows a unique thing to happen. This upper strand can now rotate in either direction, free from this part over here. Now they're still connected to the other strand by a double helix, but basically what that will do is this upper strand here will spin until the tension is relieved. Once that occurs, then the tension is lost, and now you can go back and reseal that cut. That's how topoisomerase will work. Now, topoisomerase works through if you think of topoisomerase, it is a large molecule, but it has as its functional area a tyrosine. Now, the tyrosine is also larger, and if you've forgotten what it looks like, you can look it up. But a main part of that tyrosine is a hydroxyl group. Now, this hydroxyl group here is very important for how topoisomerase works. Just so you're saying, the topoisomerase consists of a tyrosine, and part of that tyrosine is the hydroxyl group. Now, the hydroxyl group is important because what will happen, again, the hydroxyl group is important because what will happen is in that strand that we had, let's say it was a complete strand at this point, with our hydrogen bonds, the hydroxyl group will come in, and remember, oxygen is electronegative. Okay. And as that hydroxyl group comes close to the backbone, what will happen is there's an attraction between oxygen and some of the electrons here. And what that does is it actually creates a bond here with the oxygen. But now the hydrogen that was connected to the oxygen doesn't have anywhere to go, so it finds a partner in the other strand. That's how you create a cut. So it would look something like this. Then you have the backbone connected to the oxygen, and that would be connected to the rest of the tyrosine. Here's the other strand of the DNA. And here's the hydrogen. And again, they're connected by hydrogen bonds, the two strands of the DNA. Okay. But now notice this hydrogen is the only thing connected to the backbone of the DNA. The hydrogen now takes the place of the bond that would have occurred between the backbone and the other strand over there. That's what leaves this strand free to rotate. All right, so now we have the role of helicase and topoisomerase. The next enzyme that is important, kind of adding detail, is primase, the one up at the top here. Primase is needed to make an RNA primer, as indicated there. RNA primer, please note that this is RNA. Remember, we are synthesizing DNA. So in theory, everything should be DNA, but this is RNA. Okay. Now that'll have consequences later, but right now you are creating a RNA primer. Now what is a primer? It's a starting point, okay? This is the initiation area whenever you're going to create a new strand of DNA. It provides the starting point to which DNA nucleotides can be added, okay? So you can think of this part here as being DNA, okay? But the primer is needed to add the DNA nucleotides. Why? Why is a primer needed? Well, because the enzyme that creates the new strand is incapable of kind of starting a strand. It's really good at adding on nucleotides to create a strand, but it isn't good at just from nothing starting a strand. So it has to have something to start with. That's where the primer comes in, and the enzyme primase that will do that. Now in bacteria, there is a DNA polymerase called DNA polymerase III, which again is the enzyme that's going to create the new strand. Remember, synthesis only occurs in the 5' to 3' direction, and for the leading strand is going to be continuous. This is the enzyme that's going to require a starting point or a starting molecule. Now, in the case of when the DNA polymerase cannot create a continuous strand, the lagging strand is creating, so that's the discontinuous or partial strands. If you remember the previous figure, when you create a lagging strand, you have a lot of small little pieces because you have to basically start over every time the replication bubble opens up a little bit more. So you end up creating fragments, and that is shown on the bottom part here. Okay. So if you look, creating 5' to 3', notice here the arrow is going in this direction, so you are synthesizing in that direction. When the next part of the fork opens up over here, you have to start again over here with a new primer, which is shown in yellow. And that again proceeds in this direction until you hit the primer of the previous synthesis. Okay. At that point you get fragments, and those are referred to as the Okazaki fragments. Now there's another version of the DNA polymerase enzyme in bacteria. It is called DNA polymerase I. Its job is to now, once you have, for example, Okazaki fragments that we just saw in the previous slide, one fragment is going to lead up to the next one. Okay. But remember, at each fragment there's going to be a primer. Again, here shown in yellow. But those are made out of RNA. Those are eventually going to have to be removed because you don't want RNA in the middle of your DNA molecule. So it recognizes the RNA, removes it, and adds DNA nucleotides in its place. It will do this on both the leading and the lagging strands. Now you may be saying that, hey, the leading strand doesn't have these little fragments. But remember, that's only for one little replication bubble. Remember, much earlier in the DNA replication tutorial part 1, there are many replication bubbles in eukaryotes, at least. And so what it may happen is you're going to end up with fragments at one point or another. Now, this DNA polymerase is in bacteria, but I will talk later about the DNA polymerases in eukaryotes. The next enzyme we have to look at is DNA ligase. DNA ligase will now fix any little gaps that are, or breakages in the sugar phosphate backbone of the DNA molecules. Remember when you have the primers here, something will, like DNA polymerase 1, will replace the RNA nucleotides with a DNA set of nucleotides. However, that doesn't mean it will seal up those little breaks that occur, like you can see right in there, that occurs right before the primer. You need a different enzyme, which is called DNA ligase, that will come in and seal the sugar phosphate backbone together by creating the bond between the sugar and the phosphate. Now, there is a very big difference here when you replicate circular, which is bacterial, versus linear DNA. Circular chromosomes don't have an end, so there's no problem with coming to an end. Okay. But with the linear DNA, there obviously is an end, so what happens when you get to the end? The ends of DNA are called telomeres. Those are the tops and bottoms of the chromosomes. Those are the endpoints of a linear DNA. Now, telomeres, as we'll see in a minute, have the potential for becoming shorter, shortening over time. It is thought that shortening of telomeres is associated with the symptoms of aging. Okay. Telomeres that don't shorten, in some cases, appear to be associated with a different problem, which is cancer. Now, why might telomeres shorten? What's the problem with ends that might cause telomeres to become shorter with time? Okay, if you look at this particular slide, this kind of shows why telomeres will have a problem. And the problem will be on the lagging strand. So here we have replication occurring. Notice that you have a leading strand and a lagging strand. And we're getting towards the end of the DNA. So the helicase will open up the last little part of the end, and now the upper strand, which is the leading strand, will have the DNA polymerase continue completely to the end and synthesize the entire leading strand. However, with the lagging strand, remember when helicase opens up the last area, this is the lagging strand, so a new primer will have to be made, and they show that there. Primase will add the new primer, and then in the next one here, DNA polymerase will take that primer and add DNA strands to that. But notice it doesn't get all of the nucleotides at the end. The gray ones there remain there's no primer associated with it, and there is no DNA nucleotides as yet with it. In the end, that area does not end up being replicated. Well, why not? Why doesn't the primer kind of get all of these strands in there? Well, because the primer is an enzyme, and think of it this way, it needs to have something to sit on, a template strand to sit on, in order to make the primer. In order to get those kind of last little nucleotides there, it would have to have something behind it, over here somewhere, to sit on so that it could make a primer at those nucleotides. Because the DNA strand has ended at that point, there is no template strand for it to kind of hang on to. So it has to grab a little bit of the template strand, sit on it, and then create the primer farther away from the end. That leaves a part where the primer is not there. That will leave an unreplicated part of that particular DNA strand. Now, if this only happened once, maybe that wouldn't be such a bad thing. But remember, DNA replication happens every time a cell goes through mitosis or meiosis. Imagine if you were a skin cell. Remember, your skin cells are continuously dying and being sloughed off. And their skin cells have to constantly go through mitosis in order to replace the dead cells. Well, imagine if you lost DNA every single time. Imagine here there's only a small area that is unreplicated. But if that was unreplicated, then the next time this part wouldn't be, then the next time this part, and so forth and so forth, you'd get a part where you would be missing a significant part of the DNA. So this becomes a problem if it's left this way. So what can the cell do? There is an enzyme called telomerase that helps to solve this problem. The enzyme is a composite of proteins and its own RNA template, which, they have there. Okay. The own RNA template, or the template within telomerase, is represented in yellow here. Now, what is its function? It will hang on. This is the template strand. And what it will do is it will hang on to the end of that template strand. And because it has its own RNA template, what can happen is DNA nucleotides can then base pair with the template inside the telomerase. It will add nucleotides now to the template strand. Now in this particular case, the telomerase, once it's adding those nucleotides by base pairing to the template within the telomerase, this is essentially reverse transcription. Okay. You are creating a DNA molecule with an RNA template. Now, from this, you are creating essentially a nonsense strand. So notice that here the template inside the telomerase has a repeating sequence, CCC, AA, U, CC, C. Okay. What that'll allow is, notice the CC here and the CCC here will allow a unique thing to happen. As the telomerase moves down the template, so for example, it will allow nucleotides to add here as we're seeing, okay? And so what will happen is at some point a GGG will be added in the DNA strand there. Now when the telomerase picks up and moves over here, this GGG was originally associated with these C's, but later they can be, as the telomerase moves, will be associated with these C's. With that repetitive mode now, with these C's associated now with this, the rest of the template in the telomerase can now be used to add new DNA nucleotides. This has the added benefit now of, once you have created an extended template. So now you have extended the template. Now the RNA primer can start on that extended template and now be used to create the DNA or to replicate the DNA strand in this particular direction normally. Now because remember this is an extended template, here they're showing you the important difference. The gray over here was the original DNA. Because you have created the primer on the extended template, now by the time you get to the original template, which is over here, your RNA polymerase is already well on its way. Sorry, DNA polymerase. The DNA polymerase is already on its way making the new strand. Now, DNA polymerase 3 in bacteria has some important qualities. One, it is very accurate in synthesizing a DNA strand. It also has proofreading ability. It can recognize mistakes. Essentially, when it's adding a new nucleotide, it can take a look at the one it previously added. If it made a mistake, it can stop, pause the replication, removes the mistake, and then continues on. This is part of the reason the ability to remove the mistakes is the reason why 5' to 3' replication is necessary. That's why this new strand has to be synthesized in the 5' to 3' direction. In brief, the reason why is if you were to go in the opposite direction. For example, if you were to synthesize the new strand in the 3' to 5' direction, because of the way this is, the nucleotides and the DNA strand is organized, if you synthesize the new strand in that direction, the energy used to create this molecule, remember you're building something up so you need energy, would be in the actual strand you're creating. Okay. Energy is in the strand, the new strand you're creating. Okay. So if you make a mistake, so imagine if you have a strand and the energy, so the energy in the form of phosphates is in the strand. You add a new nucleotide. That takes away the energy. So now, from that, and you add a new nucleotide, so here's your new nucleotide. But that now has the energy for you to add another one. But let's say this one was wrong. If you removed it, then you would be left with a strand with no energy. So now, synthesizing in the 3' to 5' direction would work as long as you never made mistakes. But this is life, living organisms, biology. Mistakes happen. Okay. So if, on the other hand, you synthesize in the 5' to 3' direction, as is what happens, the energy is in the nucleotides. So, for example, if you're going to add a new nucleotide, the energy is in the nucleotide. So you add it, and the energy is now lost, but you get a new nucleotide. But because the energy was here, if something happens, remember the energy is not in the strand here. So if this was the wrong nucleotide and you took it off, you are still left with the same strand, and you can have a new nucleotide with the energy, add right on. This is why synthesis in the 5' to 3' direction occurs. All right, now what happens, for example, when you have errors? Errors in DNA replication. Okay. As I've just explained, it's important to be able to make sure that any errors don't persist. Now, some errors will always get through, but the body has an amazing ability to repair and to fix problems. Now, one of the ways that they can recognize problems is by a enlargement of the DNA at a place where there is a problem. Okay. The problem here, if you notice, because the two do not match correctly in their hydrogen bonding, T in DNA does not normally pair with G. You get a malfunction of the hydrogen bonding. And that creates kind of a looseness there in the hydrogen bonding, and that creates kind of like a little enlarged area where it kind of puffs out a little bit where there's a mismatch. Okay. Now what can happen is enzymes that detect this mismatch can remove the nucleotides where there is a problem, and notice it will occur around the problem. And it can then replace the missing nucleotides properly, and detecting here now a proper orientation of the DNA. Now there's one last part that we need to discuss. Here we were kind of focusing on bacterial enzymes, DNA polymerase, but eukaryotic cells use a slightly different nomenclature for the DNA polymerase. So in eukaryotes, there is alpha. Now these are all DNA polymerase, alpha DNA polymerase, and I'm just going to abbreviate it there. Now this is the one that's going to pick up from the primer and start to synthesize the DNA strand. So this adds nucleotides to the primer. But the alpha DNA polymerase is not very good at correcting errors, and it's not as efficient at preventing errors from occurring. So in eukaryotes, once alpha has proceeded and started making a DNA strand after the primer, two different polymerases will take over, which are much better at being accurate and to prevent errors from occurring. But if there is an error, it's better able to correct them. So the first one is delta, and this takes over for alpha on the lagging strand. Epsilon will take over for alpha on the leading strand. Now, one way that you can remember this is, remember the leading strand occurs faster. Okay. So in the alphabet, think of delta as D, epsilon as E. Which one is farther along in the alphabet? Well, that would be E. So that's the one, epsilon will match with the leading strand because they're farther along. Delta and the lagging strand are slower. Okay, now this is going to be the end of the second tutorial for DNA replication. If you have any questions, we can address any questions you have on any part of replication in lecture.